Hello, this is Miss Dagenford, and we're going to continue our discussion on Mendelian genetics. And what we're going to look at today is we're going to look at the variations that we have learned about since Mendel's times. Now, Mendel examined traits that had what we called incomplete dominance. Now, complete dominance means that if you have the dominant allele, you're going to show the dominant phenotype. All you need is one, and that trait is completely dominant. So we saw that with our black labs. If they had just one dominant allele, they're going to be producing black fur for the black lab. But what about variations? What if both alleles are dominant? Well, we call this codominance. And so codominance is when you see both alleles being expressed. Now, one of the more interesting examples would be in coat color in a lot of animals. In a lot of animals, they can have two different genes that are getting expressed equally. And so we call this codominance, not complete dominance, but codominance. And that makes sense. They're codominant. They both get expressed. Now, because they're both dominant, we use different notations to show this. What we do is we choose a different letter to kind of represent the gene. So in this case, we use the letter C for color, and then we might choose the letter W, capital W, for a white fur, and then the capital R for this reddish brown fur. And so in codominance, you can get what's called a roan color. This works in horses, cows, in some traits in dogs, even in guinea pigs, where if you have one of each allele, you're going to get both the reddish brown and the white. Now, from afar, this kind of makes it look like this dilute, milky brown, white brown. But if you were to get up close, you would see that there were white hairs and reddish brown hairs. So they have both of them. So let's be very clear that they did not blend. They're not making a pinky brown fur. They're making white fur and reddish brown fur right next to each other, and then we see it as this roan color. So those are the alleles, and let's take a look at our genotypes. So if we have homozygous W, CW, CW. Now that, even though it's a superscript, we count that as one letter. So CW is one allele. And so if you have CW, CW, that would be an animal that has both white fur alleles. That would be homozygous. And you'd have a cow or horse that is white. If you have CR and CW, that would be heterozygous. And because they're codominant, they're going to make red fur and white fur, and that's going to come across as a roan coloring. And then if you have CR and CR, only red fur is made. Now I say red, but it's like this reddish brown up here, you can see. And so that's the notation that you'll most commonly see with codominant traits. So let's go ahead and work one out. And we can do this one together. And then I'm going to have you do the one on the next slide yourself. So let's do where one parent is this reddish brown, and then the other parent is a white horse. So we've got a reddish brown horse and a white horse. And so let's take a look at this. So we can say we have a white CW, CW, and then the red CR, and then let's drag another one, CR. Now, just as before, the Punnett squares are no different. So you can, we can make our Punnett square a little bit bigger, and let's drag and drop And of course, just always keep track of where you're going to see everything. I believe I made plenty this time. So there's our CR. And they're going to go right next to each other. And there we go. Now, again, when you are reporting your genotype and phenotype, you always want to be using either a fraction or a ratio or percentages. So in this case, we're going to have 0 reddish brown because you can only have reddish brown if you have 2 R's. We don't have any that have that. You're going to have 4 out of the 4 that are roan, and so that would be our phenotype. And then you could also say 4 out of 4 heterozygous, and that would be our genotype. And then we have 0 out of 4 for the white. And so those are our fractions. You can also say 100% if that works for you. So let's try your hand at this one. You guys can help each other out in class. And then Zoom, what would we have? What would we set up if you have a parental cross where you have one roan horse and another roan horse, and you can set it up to show this by you can hit pause and you guys can work it out together in class. So if you did it together in class, this is what you probably got. So we now have one box that shows CW, CW, one box that has CW, CR, another box that shows CW and CR, and then one box that shows both CR. And so, again, you guys can discuss what does that mean about the outcomes. So you can hit pause and discuss how many of those 4 possibilities are going to be white, how many of those possibilities are going to be roan, how many of those possibilities are going to be white. Okay, so if you guys hit pause, you should have found that that bottom right should have been a reddish brown, 1 out of 4. And that also means that that is homozygous, red. You should have found that there are 2 out of 4 roan, and there's only one way to have roan, and that would be heterozygous. I can't spell, evidently. And then 1 out of 4 should be homozygous white, which would then mean it's a white horse. Now, one thing to notice, and this is one of the reasons I like fractions, because if you did it correctly, you should, if you were to add them all up, you should get 4 out of 4. Now, that happens with percentages too. So you could very easily say 25%, 50%, 25%, and you would be mathematically accurate as well. So let's go on to the next example of codominance, and that would be blood type. Now, a quick refresher here. When we're going over these letters and subscripts and superscripts, what that really means is a gene, right? And that's a DNA strand. Now, what that indicates is a code to make a protein. So let's make sure we're making that connection. That means that there is a length of DNA that is a code for making a protein. In the case of the black labs and the chocolate lab, that was the eumelanin, which gave it the either the black color fur or the chocolate lab color fur, and that is called eumelanin. That's a protein, same protein that causes our hair color, skin color, very similar. And in the case of the roans, it's a reddish melanin called pheomelanin, but it's still a protein. So here's the connection. We had a gene to make a protein, and some genes get expressed, they're dominant. Some genes don't get expressed unless you have both of them. And so the ABO blood type is another example of this. And in ABO blood type, you're making the antigen that's on the blood cell, the red blood cell. You can either make an A antigen, shown here in the little balls and sticks on our red blood cell. That would be the A antigen. You can have type B blood, that's showing the B antigen. If you have both alleles, they're codominant. You're going to express both the A and the B antigen. If you have a gene that is recessive, you're not going to make either. You either have them or you don't. So again, they're codominant, meaning that if you have both, you're going to have type AB blood. So here are the alleles. Again, we're going to choose a letter. They've chosen the letter I. I superscript A, I capital A would be type A protein. I capital B would be the type B protein. Little I means that there is no antigen made. So let's go over the genotypes. If you have IA, that's going to give you type A blood. You can also be IA little I, that's still type A blood. Remember, little I means nothing's made. Type A blood would be those two possible genotypes. If you have IAIB, that means you're going to have type AB blood. You're going to make both. And then type B blood, you have two possibilities, IB or IB little I, which remember is nothing. A little I would be type O. You don't make any antigens. Now, let's do a quick practice run. A man with type A blood, and we're going to say homozygous, is going to marry a woman and have kids with a woman with type B. So let's set it up. We'll do this one together. And then you guys can decide as a class what these look like. And then let's just also set up the next section. The next section is if we have heterozygous. So I, and then we're going to choose the little I, and then IB, and then the little I here. So again, you guys can pause and do these together as a class and discuss what you're going to get. So we'll come back and go over these questions.